Hello, everyone. Today we're diving into a crash course on a tour of the cell, the fundamental unit of life. Our mission today is to give you a shortcut, really, to understanding the microscopic architecture that makes all life possible. We're not just going to list definitions.
No, not at all. The goal is to see the cell as this incredibly efficient, hyper-specialized city.
Exactly, a factory that never sleeps. We're going to tour this whole cellular metropolis from its ancient origins to its, complex internal logistics.
And in doing that, we'll see exactly how the structure of these tiny parts dictates the function of everything alive.
It's kind of amazing to think that for almost all of human history, this entire world was just invisible.
Completely. It was only about 350 years ago that we even started to realize that our bodies, all bodies, are made of these tiny liquid-filled rooms or compartments.
And that breakthrough came when the tech finally caught up, right? I'm thinking of Robert Hooke in 1665.
Yes, with his crude microscope, he looked at a piece of oak bark and saw these little boxes. He called them cellulae.
Which is just Latin for little rooms. So he saw the container.
He saw the container, but it was Antonie van Leeuwenhoek who really saw the life inside the container.
With a much better microscope, I take it?
Oh, much better. He looked at pond water, scrapings from his teeth, and he got so excited. He described seeing, and this is a quote, animalcules, very prettily moving.
Animalcules, I love that. He saw a hidden world in motion.
He really did. And all of those observations eventually led to this foundational rule of biology, the cell theory.
The idea being basically that all living things are made of cells.
And this is the critical part,
Yeah.
that all cells have to come from other pre-existing cells. They don't just appear out of nowhere.
Okay, so to keep exploring this world, we need to understand the tools. It's not as simple as just looking closer.
No, it comes down to two key properties. First is magnification, just making the image bigger.
Simple enough.
But the second one is the real challenge, resolution. That's the clarity. It's the ability to tell two tiny objects apart, to see them as separate things.
So if a light microscope, an LM, is like a powerful window where we can see living things swimming around, like a paramecium, what does an electron microscope, an EM, show us?
The EM is a total game changer. Its resolution is just vastly superior. It doesn't use light, you see. It uses a beam of electrons.
And that lets you see more detail.
So much more. It reveals the cell's ultrastructure, the deep, complex internal anatomy. But, and this is a big but, the process kills the cell.
Ah, so no live viewing. That's the trade-off.
It's a huge trade-off. And with the EM, you actually get two different kinds of views.
Depending on what you want to see, the outside versus the inside, maybe.
Precisely. We use a scanning EM, or SEMA, to study the surface architecture. It gives you these stunning, almost 3D images of, say, the cilia on a cell's exterior.
Okay, and for the inside?
For that, you use a transmission EM, a TEM. It slices right through the cell, letting you see the internal details of the organelles.
So let's move on to the actual blueprints for cells. Now, no matter how different they look, all cells share some basic features, right?
Four fundamental things, yes. They all have an outer plasma membrane, they all have chromosomes with DNA, they have ribosomes to build proteins, and they're filled with cytosol, the jelly-like fluid.
But from there, they diverge into two main types.
Right. You have the prokaryotic cells, the bacteria and archaea. They're structurally a lot simpler and smaller. Their DNA is just concentrated in a region they call the nucleoid.
But it's not closed off.
Exactly. There's no membrane around it. They just lack most of the specialized membrane-enclosed compartments we're about to get into.
Which brings us to the eukaryotic cells. So that's us, plants, fungi, everything else.
Yes, and they evolved from prokaryotes about 1.8 billion years ago. The defining feature is a true membrane-enclosed nucleus for the DNA, plus a whole host of other membrane-enclosed organelles.
It's like different departments in the city.
That's a perfect analogy. And every city needs a border. That's the plasma membrane. It's the ultimate traffic cop.
And it's made of this thing called a phospholipid bilayer.
Right. It's a clever arrangement. The heads of the phospholipids love water, so they face outward toward the watery environment inside and outside the cell.
And the tails hate water.
They're hydrophobic, so they point inward, creating this oily barrier that separates the two watery environments.
But it's not a solid wall. There are proteins embedded in it, acting like gates and pumps.
Yes, that's the traffic cop part. They form channels that strictly regulate what comes in like nutrients and what goes out like waste.
This need to move things in and out actually creates a limit on how big cells can get, doesn't it? The whole surface to volume ratio idea.
It's a crucial concept. As a cell gets bigger, its internal volume grows much, much faster than its surface area.
So the bigger it gets, the less membrane it has relative to its insides.
Exactly. And that membrane is the only way to get nutrients in and waste out. If a cell got too big, it would literally starve or poison itself because it couldn't service its huge internal volume efficiently.
So staying small means you have a high surface area compared to your volume, which is much more efficient for life.
That's the key.
Okay, let's go inside the eukaryotic cell now, starting with the control room, the nucleus.
City Hall, for sure.
Right.
This is where the genetic master plan, the DNA, is housed. And from here, all the major operations are directed, especially making proteins.
And inside, the DNA isn't just floating around, it's packaged with proteins into something called chromatin.
Right. And the whole thing is protected by the nuclear envelope, which is actually a double membrane that has pores all over it.
And those pores aren't just open doors, I assume.
No, they're highly regulated. They let specific instructions out, but the master blueprint, the DNA itself, stays safely inside.
And what's that dark spot you always see inside the nucleus? The nucleolus?
That's the ribosome factory. It's a dedicated site for building the components of the very machines that will go on to build proteins.
So the whole information flow is DNA instructions are copied into messenger RNA, or mRNA.
Which then leads the nucleus through a pore, carrying the instructions to the factory floor.
And that factory floor is littered with ribosomes, the assembly workers.
The actual machines, yeah. They read the mRNA instructions and build the proteins. A cell that makes a lot of protein, like a pancreas cell making insulin, will have millions of them.
And they come in two flavors, right? Free and bound.
Correct. And it all depends on where the protein they're making is going to end up. Free ribosomes just float in the cytosol, and they make proteins that will be used right there in the cytosol.
Like enzymes for breaking down sugar.
Exactly. But the bound ribosomes are physically attached to another organelle, the ER. They make proteins that are destined to be put into membranes or packaged up and shipped out of the cell entirely.
Okay, so that leads us right to the endoplasmic reticulum, or ER, the biosynthetic workshop.
It's a great name for it. It's this huge network of sacs and tubes, and it's actually physically connected to the nuclear envelope. It's the largest membrane system in the cell.
And it also has two distinct regions. Let's start with the smooth ER.
Smooth, because it doesn't have ribosomes on it. It focuses on non-protein jobs. Two big ones to remember. First, lipid synthesis. It makes oils, phospholipids, even steroid hormones.
And second,
detoxification. This is huge in your liver cells. The more you expose your liver to certain drugs, the more smooth ER it actually builds to cope. That's what leads to increased drug tolerance.
Fascinating. So if smooth ER is for lipids, the rough ER, which is rough because it's studded with those bound ribosomes,
is the protein modification and shipping department. As the ribosomes build a protein, it gets threaded directly into the ER's internal cavity to be folded and modified. The rough ER also acts as a membrane factory, building new membrane for the cell.
And from the ER, these products move on. Where do they go next?
They get packaged into little bubbles called vesicles and sent to the Golgi apparatus. If the ER is the factory, the Golgi is the central post office and quality control hub.
And it looks like a stack of pancakes.
A stack of flattened, unconnected sacs, yes. And it's a very organized process. Vesicles arrive at the receiving side.
So no way.
The products inside are processed, modified further, and this is key. They get molecular tags attached to them, sort of like zip codes.
To make sure they get to the right destination.
Precisely. Then they're repackaged into new vesicles that bud off the shipping side,
Wow.
headed for their final location.
All right, what about sanitation and storage? The city's cleanup crew.
That would be the lysosomes. Think of them as the cell's recycling centers. They're basically little bags full of powerful digestive enzymes kept in a very acidic environment.
And they break down. What?
A few things. They can digest food that the cell has ingested, but maybe their most important job is something called autophagy.
Self-eating.
Literally. They engulf and break down the cell's own old damaged organelles. It's critical maintenance. If those enzymes are missing, like in Tay-Sachs disease, this undigested junk builds up and severely damages the cell.
And for more general storage, we have vacuoles.
Right. They're basically just large vesicles. In plants, the central vacuole is enormous. It stores water, chemicals, pigments, waste. Its expansion is actually what makes the plant cell grow.
So we've covered manufacturing and logistics. Let's talk about the power grid, the energy converters.
The power plants of the cell. First up, mitochondria. You find these in nearly all eukaryotic cells. Their job is cellular respiration,
Taking the energy from food and converting it into ATP.
the universal energy currency of the cell. And their structure is perfectly suited for this. They have two membranes and the inner one is highly, highly folded.
Into those structures called cristae, the folds are to increase surface area, right?
Massively increase it. More surface area means more space for the enzymes that make ATP. It's all about maximizing energy production.
Then in plants and algae, you have another power plant, the chloroplasts.
Their job is photosynthesis, converting light energy into the chemical energy of sugar. They have their own internal structure of stacked sacs called grana, which is where chlorophyll traps the sunlight.
And having these two distinct power systems inside one cell leads to one of the most amazing ideas in biology.
The endosymbiont theory. It's a fantastic story. The theory proposes that mitochondria and chloroplasts were once tiny, free-living prokaryotes.
And they got swallowed up by a larger host cell.
Exactly. Engulfed, but not digested. They formed a partnership, a symbiosis, that was beneficial for both.
What's the evidence for something like that? It sounds pretty wild.
The evidence is incredibly strong. Both mitochondria and chloroplasts have their own single circular DNA molecule, just like a prokaryote. They have their own ribosomes that resemble prokaryotic ribosomes, and they reproduce on their own inside the cell.
Wow, so they're like ancient tenants that never left.
And became essential parts of the household. The first event was probably engulfing an oxygen-using prokaryote, which became the mitochondrion. That happened in the ancestor of all eukaryotes.
And later, one of those cells engulfed a photosynthetic prokaryote,
which became the chloroplast, leading to the plant and algae lineages, an ancient partnership.
Okay, let's wrap this tour up with the cell's internal scaffolding and its external connections, the cytoskeleton.
Right. This is a whole network of protein fibers that provide structural support, but also acts like a railway system for movement inside the cell.
It's not static, it's dynamic.
Very. It has three main types of fibers. The thickest are microtubules. Think of them as hollow tubes. They act as tracks for organelles to move along, and they're essential for separating chromosomes during cell division.
Then you have the thinnest ones, microfilaments.
Made of a protein called actin. They're critical for holding the cell's shape, especially in animal cells that don't have a wall, and they are the engine of muscle contraction, working with another protein, myosin.
Okay, and what about outside the cell? Animal cells have the extracellular matrix, or ECM.
Yes, it's this sticky layer of glycoproteins, like collagen, that helps hold cells together in tissues. It also communicates with the inside of the cell, influencing cell behavior.
And how do cells in a tissue actually connect to each other?
Through specialized cell junctions. You have tight junctions that basically stitch cell membranes together to prevent leakage, like in your digestive tract.
Like a waterproof seal.
Exactly. Then anchoring junctions are like rivets, holding cells together in strong sheets, like in your skin. And finally, gap junctions are channels that directly connect the cytoplasm of two cells.
For communication.
Instant communication. It allows ions and signals to pass between them, coordinating activity, like in your heart muscle.
And for plants, they have the rigid cell wall for support. What's their version of a gap junction?
They use plasmosmata. These are channels that pass right through the cell walls, connecting the cells and allowing them to share water, nutrients, and chemical messages.
So we've completed our tour of this incredible cellular city. What is the main, the core insight here?
It's the synthesis. It's seeing how it all works together. We looked at 4 functional groups, really. Genetic control, manufacturing and logistics, energy processing, and then support and communication.
And none of them work in isolation.
Not at all. The truly amazing thing we call life emerges from the seamless, coordinated interaction of all these parts. The cell really is so much greater than the sum of its parts.
And that actually leads right into our final thought for you. Think about that interdependence, especially in a muscle cell. For a single contraction, you need the smooth ER to release calcium, you need microfilaments to do the actual pulling, and you need mitochondria churning out ATP to power it all.
Right. A perfect storm of cooperation.
So imagine this, a single genetic mutation that only affects the motor proteins that interact with the cytoskeleton. How could a failure in just one component of that support system cascade through all 4 functional systems and devastate the entire organism? That incredible level of interaction and the domino effect of failure. That's really where the complexity and fragility of life resides.